You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Dr. Ben Long from the Australia National Union College of Science, National University College of Science. Dr. Ben graduated with a PhD in plant biology from La Trobe University, working on toxic cyanobacteria, which is blue-green algae. He's done a lot of postdoc work and looked at cyanobacteria and the various compounds involved with them, which we'll get into. So, Ben, thanks for coming. How you doing?
Good. Thanks, Richard. Thanks for having me.
Yeah. So, first question, why the fascination with cyanobacteria and the compounds associated with them and with plants? What kind of drives your interest?
Yeah, well, it's a really good question. I started off as an undergraduate student just studying biochemistry and plant biology, and had the opportunity to work on cyanobacteria as a research student, and it was just something a bit different and a bit simple compared to most plants. I mean, plants are really complicated organisms that can grow, hundreds of yards tall. But cyanobacteria, you can grow them in a glass tube, and it makes them simple to work with. And I just thought they were great fun and really simple little systems to get an understanding of biochemistry. So that's where I started with them. And they've taken me across a lot of fields of travel.
So what kind of things have you studied about cyanobacteria that you found to be particularly interesting or amazing or unusual?
Well, the first thing that I did with them is work on them as to look at how they produce toxins. Most of the cyanobacteria around the world that people know of produce toxic blooms, and I wanted to understand the toxins that they produce. And over the years that I've been working on them, it appears they produce more. There are more and more compounds that we're finding that they produce that tend to be toxic or have some sort of strange bioactive characteristic. And this kind of thing is just popping up all the time, which I find is really amazing. And in the last 5 to 10 years, for example, we've found that cyanobacteria seem to produce a strange amino acid called BMAA that turns out may play a role in neurodegenerative diseases, which is really quite interesting. So there's just all these amazing things that they produce that seem to play a role in our everyday lives that we're only just finding out. And I think that's what's really cool about them.
I thought I had read that it was theorized that cyanobacteria integrated themselves with a prokaryotic cell to create plant cells, countless millions or billions of years ago. Is that right?
Yeah, that's exactly right. And that's one of the reasons that there's a kind of a link between what I do and plant biology. So hundreds of millions of years ago, there was an organism like an amoeba, the typical diagram that you've seen in a Larson cartoon, those wobbly little organisms that might be playing guitars or something in one of these cartoons. They tend to eat things by a process called phagocytosis. They basically gobble them up, wrap themselves around them, and they dissolve whatever they eat. But on a number of occasions in evolutionary history, this has happened, and the organisms that have been eaten have actually managed to stay alive inside these amoeba-like organisms and replicate. And so that's what happened with a cyanobacterial cell. and it turned into an algal cell that we know today that then also evolved into land plants. So there's these amazing abilities that a lot of organisms have had to take up another organism inside their own cells, and those organisms have become what we call organelles. So the green component of a plant cell or an algal cell is what we call a chloroplast, and they started off their evolutionary history as something that was a common ancestor to our current day cyanobacteria.
And then even for animal cells, and also supposedly a mitochondria is what was engulfed to create a mitochondria? What kind of a cell?
Yeah, so that's the same kind. Yeah, that's the same kind of story. It was some kind of ancient bacterial cell that was taken up and turned into what we now call a mitochondrion. And again, the mitochondrion is an organelle that carries out a particular process inside both plant and animal cells. And that's really the key interesting thing about the work that I do now. So one of the things we recognize about what you're talking about, eukaryotic organisms, they're plants and animals that we see as some sort of more further evolved organism that has within its cells that has packets of organelles that carry out specific biochemical reactions. And the other type of life is the prokaryotic organisms like the cyanobacteria and the bacteria that we have always a very long thought of as having no subcellular organelles. We've always thought that they had no compartments to do biochemical reactions in. But that's where the work that I do these days it's a starting point. We have recognised over the years, the last, say, 2 decades, that cyanobacterial cells in particular have these little organelle-like compartments inside them called carboxysomes where they carry out a major process of photosynthesis.
Huh. So they carry out photosynthesis in like a segregated area inside their cytoplasm, if that's the word for it, just like animal and plant cells.
Yeah, that's right. That's exactly right. Well, the specific process they carry out is the CO2 fixation component, or the capturing of carbon dioxide and turning it into simple sugars, happens inside this carboxysome structure. And it's rather than in animal or plant cells where the organelle is an ancient organism that was bound by a lipid surrounding membrane. In these cases, in the case of the carboxysome, it's actually a protein capsule that looks very much like a virus. It's icosahedral, meaning it has 20 triangular facets, but it's surrounded by protein, and stuffed inside it at a very high concentration is this enzyme called rubisco. An enzyme is a protein whose job it is to carry out a chemical reaction at very high speed, and this rubisco enzyme is packed inside. And the way the whole system works essentially is that cyanobacterial cells are floating around in the water where they don't have access to the air like most plants do. They have to get their inorganic carbon or their carbon dioxide in the form of bicarbonate. That's essentially just a dissolved form of CO2. They can pump that into their cells at really high concentrations, and then that bicarbonate will diffuse into this carboxysome structure and generate a really high concentration of carbon dioxide around the rubisco enzyme, and that makes it work incredibly efficiently. Plants, on the other hand, they breathe carbon dioxide from the air, and so there's a relatively large amount of CO2 available to them, but they also have this rubisco enzyme, and rubisco is notoriously slow and inefficient. What I mentioned that it's essentially a protein that carries out a catalytic reaction, speeding the reaction up, but the rubisco reaction only happens at about 2 or 3 times per second in most land plants, so that there are other enzymes that carry out reactions at tens of thousands of reactions per second. So it's really slow, and occasionally can't tell the difference between oxygen and carbon dioxide. So sometimes the enzyme will capture oxygen from the atmosphere instead of carbon dioxide, and when it does that, plants have to carry out a very wasteful, what we consider a wasteful process called photorespiration to recapture the lost carbon dioxide that happens in that process, and they have to spend a lot of energy. And so all organisms that photosynthesize have to do this process of capturing carbon dioxide through the rubisco enzyme, and every one of them has had to come up with a solution to the problem that the enzyme has. And land plants have got around that problem by producing a lot of the enzyme. If you have a lot of the enzyme, then there's a greater chance that you'll get the reaction happening. But from our point of view,
a couple quick questions to maybe step back a little bit. But cyanobacteria, do they tend then to congregate around, sources of bicarbonate, like animals with shells in the ocean, snails, or do they tend to congregate around reefs or other places like that?
That's a really good question. They possibly are capable of locating themselves somewhere where their resources are more predominant. But generally, inorganic carbon is distributed quite well within the environment, except for, places, for example, like on the, marine shores where the water's lapping or waves. There tends to be a higher concentration of gases when you've got waves crashing. So there's a greater propensity sometimes to find cyanobacterial cells in those kind of areas. In stagnant water, tends to be low concentrations, and you might find that there are low concentrations there. But usually when people see cyanobacteria, it's when there's a bloom event, and they tend to be everywhere. So I think they can make use of quite small amounts of inorganic carbon when it's available, and that's really their evolutionary advantage, is that when the carbon dioxide availability or bicarbonate availability drops in their environment, they're actually able to adapt and switch on this system that we call a CO2 concentrating mechanism, so that they actually have an advantage over other organisms under those conditions. And that's one of the reasons that they are able to form a bloom, is that they can take off very quickly when the conditions are not that great for other organisms to live.
And what does a bloom look like? Is this the famous red tides, or what does it look like to the eye?
Yeah, it's similar to a red tide, but obviously green or a little bit blue-green. You often see them around the edge of a lake or a river, and sometimes they can produce what we call a scum, which can be inches thick, like a mattress on top of the water, really thick. If you put a stick in it, it'd be like sticking a stick into a pail full of paint. Very thick and gooey green material. And sometimes when it dies on the shoreline, you can see a lot of blue pigment appear. That's when you know it's dying. The recommendation is if ever you see one of those, don't drink the water, don't swim in it, don't let pets go in it, because there's a very good chance that it's toxic.
And you said they produce toxins. What do they produce toxins in response to?
Yeah, that's a really, really good question, and it's still a mystery despite the fact that we've been working on these kind of things for decades now. There's a particular species of cyanobacteria called Microcystis aeruginosa, which produces a toxin called microcystin, and we really don't know why it produces the toxin, but just as an example of how toxic it is, when it was first isolated, it was referred to as the fast death factor. It would kill lab animals in incredibly high rates, at very high speeds, at very low concentrations. But despite working on it, since the 1950s, people still haven't pinned down exactly what the cell's producing it for. We know that particular toxin is produced all the time.
Yeah, I would think that they would produce certain things when they're blooming and when they're not, and then in low carbonate environments and high. I mean, I would think all this is in response to environmental pressures, right?
Yeah, well, we've the PhD project that I worked on, in fact, was looking at exactly that, trying to work out under what growth conditions we find the cells producing more or less of this particular toxin called microcystin. And essentially, the bottom line is that we found that the faster the cells grow, the more toxin that they produce, but there doesn't seem to be any deliberate switch that turns the toxin production on or off. It's always on, regardless of what conditions the cell's growing in, unless there's a different genetic type of the organism swimming around in the water. Sometimes the organism doesn't have the genes to produce the toxin. but the one that does have the genes will always produce it, and we don't know why. And it's a really good question that you ask. It's exactly the question that everyone thought. Well, there must be something controlling it, and we can't really pin it down despite decades of work.
Well, what does it do to its local environment? Does it kill fish? There's got to be a reason that it produces it, or maybe it's not even a toxin if it's produced in its local environment, but it's only a toxin to things on land. I don't know.
Yeah, that's a really good question. I think that's where we're heading with specifically with this microcystin toxin, for example, is that it plays a role in the cell for reasons that we still haven't exactly worked out, and it's toxic just by chance. It doesn't appear to have a role in doing much in the environment. It might have effect on some of the organisms that feed on the cyanobacteria, so some fish and some very, very small organisms that eat them can be detrimentally affected and by the toxin and maybe even killed at very high doses. But we don't think that's a major ecological role that they're playing. So there's not necessarily an advantage in production of the toxin for them under those conditions. It seems to be just by chance. And in fact, the toxin is toxic because it binds to another enzyme that nearly all eukaryotic organisms, all of those ones that have those packages inside their cells, they all have an enzyme called protein phosphatase, whose job is to cut phosphate groups off proteins, which is a really key programming step to make sure certain enzymes and proteins are switched on. So it stops that process from happening, which makes cells kind of constantly switched on, which is not a very good system to be in. You don't have any cellular control. And it's just by chance that the toxin has this capability. And yeah, really, we just don't know. And all of the questions that you have are questions that we're still struggling with.
Well, have you tried knocking out the sequences of RNA or whatever it is in the bacteria that code for these toxins to see what happens?
Yeah, so obviously that was a question that people wanted an answer to, seeing if they could do that, and it's been done probably nearly 15 to 20 years ago, or probably more than 20 years ago. And we certainly, people work with strains that have the toxin genes knocked out of them, and those organisms seem to behave in a very similar manner to what we call the wild type, the one that doesn't have the genes knocked out. All the differences seem to be very subtle, and the toxin doesn't seem to be a major contributor to the physiology of the organism. So, yeah, we still don't know the answer to that, and people are still looking. But I've moved on from toxic work, and I'm more interested in the CO2 capture. So I've kind of left that alone, and I'm just keeping an eye on the literature to see what kind of ideas people come up with.
Yeah, what's the goal in studying these organisms? What kind of role do they serve in their local ecosystem? And then, do people envision that they could be used in bioreactors to pull CO2 out of the air or for other uses? I mean, what's exciting about them?
Yeah, well, it's really exciting. I mean, obviously, the reason we know them is that they tend to produce these blooms that are toxic, and there's generally a negative connotation for them, and most people think that we've got to try and get rid of them. But yeah, they're a huge application in bioreactor work. For example, trying to produce oils from them that can be used as fuel, or getting them to produce other useful bioactive products. But the key thing that we're interested in is using that CO2 concentrating mechanism that I described, and we're actually trying to take that system and move it into crop plants to make crop plants more efficient at carbon dioxide fixation. That's the real key that we're working on, but there are other people working on, as I mentioned, like oil production or capture of carbon dioxide from, for example, coal-fired power plants where there's a lot of CO2 released from the plant during coal burning, that some of that can maybe recaptured into biomass through pumping exhaust gases through cyanobacterial cultures. So the fact that they can fix carbon dioxide quite efficiently is really one of the reasons that we're interested in them from a crop plant perspective.
Well, once they fix the carbon dioxide, what happens to it inside of the algae? What form does it take or stay in, and what happens to it?
Yeah, it's a good question. So exactly the same process biochemically happens to the carbon dioxide in cyanobacterial cells as does in plant cells. So essentially the first step is via that Rubisco enzyme, where carbon dioxide is essentially glued onto the molecule that's 5 carbon atoms long, and in doing so produces an intermediate molecule that's 6 carbons long that gets cut in half. So we end up with 2 simplistic sugars that are 3 carbons long. So that means there are 3 carbon atoms glued together. One of those is a new one coming from carbon dioxide from the atmosphere. So we get the production of 2 3-carbon simple sugars that then go on to produce more complex sugars inside the cell that essentially all of the components of the cell are then constructed from, and the cell can also extract energy from if it needs to. Now, that 3-carbon sugar concept is the reason that you often hear plant scientists refer to what we call C3 photosynthesis. It's the process whereby plants and all photosynthetic organisms capture carbon dioxide from the atmosphere and first turn it into something that's a C3 molecule.
Gotcha. There's a few that are C4, which I guess is a 4-carbon sugar.
Yeah, that's right. Yeah, so C4 is a kind of CO2 concentrating mechanism like cyanobacteria use, but found in plants. It uses a very complex but slightly different mechanism to that cyanobacteria use. Those plants also carry out what we call C3 photosynthesis, but strangely enough, the very first step of the process is to take bicarbonate inside the cell and use the carbon atom inside the bicarbonate, glue that into an organic acid, and that organic acid is then pumped into a special cell that we call a bundle sheath cell, and then the carbon dioxide is released again from it so that it can go through that Rubisco process, the C3 process. So that's kind of like a pump in a similar way to the cyanobacterial system. CO2 comes into the cells, converted to bicarbonate just through a dissolution, a dissolving process. Then we get an organic acid produced from that, which is then pumped across a membrane into another cell, and then that's concentrated there so that CO2 can be released and captured again. So there are actually some really exciting work being done by collaborators of ours in other universities around the world, trying to generate a C4 rice plant. So rice is currently a C3 plant, which carries out this relatively inefficient carbon capture by producing a lot of the Rubisco enzyme. And there's a real scientific push currently to try and turn rice plants into C4 plants so that they can actually capture carbon dioxide more efficiently and more effectively.
So what would be the point of turning rice into a C4 producer? Would it be more photosynthetically efficient?
Yeah, so this really comes back to the work that I'm working on and myself and colleagues at various other institutions. I'm currently funded within a program called RIPE. It's called Realising Increased Photosynthetic Efficiency. It's a real mouthful. I think someone invented the word RIPE and then tried to back translate it to something. But the whole idea of RIPE is that we recognise that there are several aspects of the photosynthetic process that are inefficient, and that those processes we can't seem to, or we haven't seemed to be able to improve them through breeding. There are other parts of crop production that we've been able to improve through breeding. For example, something that we call the harvest index, which is how much of photosynthesis and plant productivity is turned into grain that we can eat. That we managed to improve that over the last, say, 5 decades.
Well, I heard
really
that
to a large extent.
Yeah, I've heard like the average photosynthetic efficiency of most plants on Earth is like 1%, and we're hoping to get to 1.15 or 1.2%.
Yeah, it's really quite small. So there's like 3 or 4 components of if you like grain yield that the plant does that are measurable quantities. They're efficiency components. The harvest index is one of them, and the efficiency of photosynthesis is another. Things like harvest index have improved to what we nearly at what we know are their theoretical maxima. So we've nearly bred plants to the best that they can achieve in, for example, harvest index. But photosynthetic efficiency, that's the efficiency with which a plant can capture sunlight and capture carbon dioxide and turn it into crop biomass, like grain. We know that there are theoretical limits to that we're nowhere near. So we can probably improve that 3 to 4-fold on what we can get right now. So we're really at the base of that. And interestingly, what I think is really quite interesting is that we haven't been able to improve any of that through breeding. And we realise that's essentially because evolution has locked crop plants into a certain process of photosynthetic CO2 capture from the atmosphere that has limitations that it can't get around through evolutionary improvements that you would get through breeding programs.
Are you able to evaluate the ideal efficiency based on the chemistry? Like, the Carnot cycle for engines has a theoretical max efficiency. Has that been calculated for photosynthesis?
Yes, exactly. Colleagues of ours have done that. Unfortunately, I'm not a modelling person, so that's not I'm not that smart on it. So let me just put it that way. But definitely, you can break down the expected efficiencies of all of the processes within photosynthesis. And photosynthesis itself, the capture of light energy and carbon dioxide, we know how efficient that should be mathematically, and we know that we're not achieving.
Yeah, what is that? Otherwise, there'd be no, they wouldn't bother. They would just give up. So what is, what the theoretical maximum is?
I think, don't quote me on it, I don't have the numbers in front of me, but I think that, if you think of all of the components that lead to crop yield as units that add up to 1, so harvest index, for example, is, how much we can put into the grain based on how much plant biomass there is, has an upper bound of a particular value, and we're at about 80% to 90% of the upper efficiency value for that. But for CO2 capture, for example, and sunlight capture, we're only at about 10 to 30% of the upper limit of that value. And there are some other components of that yield calculation where we're up to closer around about 90% of the expected maximum. So we know that there's that gap with the photosynthetic efficiency that we could improve through other techniques other than using crop breeding, because we know crop breeding hasn't worked. So that's really where we're working. And there are a number of biochemical factors within that photosynthetic process where we know that there are bottlenecks that we can improve. One of the key ones is the fact that crop plants use C3 photosynthesis, and that they're kind of locked into making the choice of producing a lot of the Rubisco enzyme in order to capture a lot of CO2 from the atmosphere. And that bottleneck means that they have to use a lot of nitrogen to produce a lot of that protein Rubisco. But from our point of view, we would prefer that nitrogen was put into the grain, as an example. It also means that most crop plants have to breathe a lot of the atmosphere, and in doing so, just capturing 1 molecule of carbon dioxide from the atmosphere means that they lose something like 400 molecules of water. So there's really efficiencies in water use is a major problem with crops, because just breathing in carbon dioxide means that you've got to breathe out a lot of water. C4 plants, however, for example, they're able to fix carbon dioxide from the atmosphere much more efficiently and lose a lot less water. So there's one advantage there to the C4. That's why we're trying to, for example, put the CO2 concentrating mechanism from Cyanobacteria into plant cells. It's kind of like a single cell form of the C4 mechanism, which we would hope enables them to use less nitrogen and less water to fix perhaps more carbon dioxide.
Well, why not just co-locate them, where you're going to have a field somehow, have a bunch of cyanobacteria and ponds around it or near it, or maybe is making up, maybe you have a huge hydroponics operation and you have cyanobacteria in the water medium, so they produce a lot more CO2.
That would mean that there'd be a lot more CO2 capture, but we wouldn't be contributing to crop yield. So really the key here is to try to improve crop yield. And if people, for example, are eating rice or potatoes or maize or corn, for example, then that's what we want to eat. So that's what we want to be able to produce more of. So the idea is to try to incorporate that CO2 concentrating mechanism or a synthetic version of it into a plant cell so that the plant itself can capture more CO2 from the atmosphere and actually turn that captured carbon into something that we can eat or into useful biomass.
So what do we know from C4 plants? Like, where do they tend to be, and why do they have a different method of photosynthesis? Like, what, again, it's probably all environmental.
Yeah, that's a really good question, and you're absolutely right. It is environmental. It appears that most C4 plants tend to have appeared in evolutionary history in a lot of hot, dry areas, and that's kind of driven by the fact that I mentioned that C4 photosynthesis seems to be a lot more water use efficient. So plants with C4 physiology have the capability of fixing carbon dioxide from the atmosphere without spending as much water. So that gives them an evolutionary gain in hotter, drier climates. So over several million years, we've seen that C4 photosynthesis has actually popped up quite a number of times in a lot of unrelated plant species, and it's nearly always in hotter, drier areas. I don't mean desert areas, but hot, warm, tropical areas where they tend to be favoured. But we also see other interesting ways of photosynthesizing in deserts, for example, where there are other efficiencies to be gained in trying to trap water and maintain water within a photosynthetic leaf.
How much more efficient is a C4 plant versus a C3 one?
Yeah, you've stumped me there. I don't have the numbers in front of me, but they are much more efficient. And a really easy way to tell is if you go to a sugarcane farm or even a maize or corn farm, you walk into the crop, you can immediately tell you're amongst C4 plants. A lot of C4 crops are grasses, but they're not like the grass that you have on your lawn. They're 4 to 10 feet tall, and that's kind of an indication of their relative efficiency. They're able to grow incredibly fast and incredibly tall, despite the fact that they're grasses, because they have this C4 mechanism. They're really efficient at capturing carbon dioxide from the atmosphere compared to their C3 cousins.
Did C3 come before C4 evolutionarily, or do we know?
Yeah, so we think that C3 came first. So C3 really is the basis of all carbon dioxide capture. So C3 really just describes the capture of CO2 in the initial step through the rubisco enzyme into a C3 molecule. Now, everything that captures carbon dioxide through the photosynthetic process has to do that. And Cyanobacteria were amongst the earliest organisms that could photosynthesize on the planet. And there's some evidence of their existence over 3.5 billion years ago on the planet. So that's really interesting because that means we had organisms on the planet that were able to capture carbon dioxide from the atmosphere probably using this enzyme rubisco or a forebear of it. But that was in an atmosphere on the planet where there was a lot of carbon dioxide and practically no oxygen. And remember I said that oxygen is the molecule that rubisco can't discern between oxygen and CO2. But because of the photosynthetic process, about 2.5 billion years ago, that process got linked to the production of oxygen as a way of driving the system. And we started to get a lot of oxygen in the atmosphere. So from about 2.5 billion years ago, we started to get oxygenic photosynthesis, production of oxygen in the atmosphere, and this enzyme was evolving as the Earth grew older. It was evolving in an atmosphere that was increasingly full of oxygen and decreasing in the amount of carbon dioxide, despite the fact that carbon dioxide is what it wants to eat and oxygen is what it gets confused with. So as evolution proceeded, every organism that was then capturing carbon dioxide from the atmosphere using the rubisco enzyme had to work out another strategy to get around the problems of the enzyme being slow and confusing oxygen with carbon dioxide. Because the way evolution works, we get branches of organisms appearing in different ecological niches over time. Different organisms were able to then, or given the opportunity, if you like, then to evolve different ways around the problem. So cyanobacteria, we think that's what drove them to evolve this CO2 concentrating mechanism and the encapsulation of the rubisco enzyme inside this carboxysome structure, whereas land plants, because they evolved in a different time and a different space, and they actually probably appeared when oxygen had a sudden burst in the atmosphere, and they were given a different struggle evolutionarily to try to overcome the problems that rubisco had. They essentially just made the solution, well, let's produce a lot more of the enzyme to increase the likelihood that we'll have a carbon dioxide fixation reaction happening. So you can see that we've had completely different ways of getting over the rubisco problem just because of differences in evolutionary time and ecological pressures. C4 plants then probably evolved because those land plants were able to move into hotter and drier environments and have that different ecological challenge.
So what does it look like if you successfully integrate blue-green algae into photosynthetic plants? What do you expect them to do, and what does the success look like, or what are some numbers associated with it?
Yeah, well, that's a really good question. Of course, we wouldn't have done any of this if there hadn't been some mathematical modelling first to tell us whether or not it was worth trying. And we had colleagues at the University of Illinois that crunched some numbers some time ago to try and work out what are the likely outcomes if we were able to put this system into a chloroplast of a plant. Now, keeping in mind that a chloroplast of a plant originally has its evolutionary origins as a cyanobacterium, but the evolution of the CO2 concentrating mechanism happened well after that incorporation event occurred, well after the construction of a chloroplast, we think. So the idea is, we have to put in two components into the chloroplast to make this system work. One of them is a pump that pumps bicarbonate into the chloroplast. And calculations suggest that just pumping that bicarbonate in could give an increase in crop yield of around about somewhere between 5 and 15%, so around about 10% improvement just by pumping more bicarbonate into the cell, because what that does is increase the relative concentration of carbon dioxide around the enzyme rubisco. The other step to the whole process is constructing this structure called the carboxysome with the rubisco encapsulated inside it. If we just put that inside a chloroplast, because of the way the cyanobacterial rubisco enzyme works, we actually would expect that to cause a problem for the plant. If we can't do that in conjunction with elevating the bicarbonate concentration by putting a pump in, then we actually expect those plants to be quite sick, and we actually find that is the case when we try to put some of these components in. That's essentially because the cyanobacterial rubisco enzyme has its own little issues, and it has evolved to operate inside a CO2 concentrating mechanism. So that kind of hints that we need both the pump and the carboxysome in order for this system to work. But then when we get the two components in a chloroplast inside a plant cell working together at the same time, then the mathematical modelling predicts that we could have around about a 30 and maybe even up to possibly 60% improvement in yield. That upper level we really need to do some more calculations on, but 30% is an enormous improvement in yield. At least a 30% improvement at least a 30% improvement in CO2 fixation. But fixation and yield aren't necessarily exactly linked. It's a lot more complicated than that.
What about looking at the natural variation of cyanobacteria, either strains or species worldwide in different climates? I would think that there isn't just one kind, but there'd be many different expressions of what they do, variations.
Yeah, absolutely. That's a really good point, and we're really only just getting into that. So interestingly, there are two completely different types of cyanobacteria out there in the world, The ones that most people see in lakes and rivers are what we call beta cyanobacteria, and they produce what we call beta carboxysomes, and they have a different evolutionary form of rubisco inside them. There's another form out there called alpha cyanobacteria, and they have a different rubisco inside their carboxysomes. And interestingly, we know a lot about the beta cyanobacterial rubiscos, because really this whole system comes down to how fast the rubisco enzyme can work. So the beta rubiscos tend to be a little bit faster than the alphas, but we've only examined the properties of a small handful of the alpha rubiscos in alpha carboxysomes. So we really need to have a look at a lot more of the organisms that are out there, because cyanobacteria are found in all sorts of ecological niches, literally on every continent on the planet, from Antarctica to North America. They're everywhere. So yeah, there's a lot of research to be done on finding out what the true diversity is of these systems. And in fact, we're finding that there might even be other organisms, for example, deep sea vent bacteria or archaea that are able to also capture carbon dioxide but without using sunlight. We're finding that those organisms have some of the components to this system that we think we might be able to hijack for our own purposes.
How much cyanobacteria is there on the Earth? Just an unbelievable amount, or?
Well, that's a really good question. There have been some suggestions that within the oceans, 25% of, well, let's put that another way, around the entire world, most of the carbon dioxide capture by photosynthetic organisms is in the oceans, and that contributes about 50% of the global carbon dioxide capture, and about 50% of the ocean photosynthesis occurs in cyanobacterial cells. So that represents about 25% of the global carbon dioxide capture. That's just a really rough estimate, but it's probably not that far off. And you also consider that within freshwater lakes and rivers that cyanobacteria are quite dominant in certain times of the year. Cyanobacteria are a major, if not the major, contributor to what we call global primary productivity or CO2 capture from the atmosphere, comparing them to other photosynthetic organisms. So they're really, really very important.
Well, and I guess all I've done is really open up 8 million more questions as to, what could be done here. But it's been an interesting talk so far.
Yeah, it's great. Well, thanks very much.
Yeah. What are some places for listeners to get resources to learn more?
Well, the RIPE consortium that I work for, Realising Increased Photosynthetic Efficiency, people can look that up. They just have to Google RIPE photosynthesis. I also work as part of the Australian Research Council's Centre of Excellence for Photosynthetic, or for Translational Photosynthesis, and that's a bit of a mouthful, but if you follow your links or look for photosynthesis.org.au, that might give people a lot more information about the kind of work that we do.
That's great. Well, Ben, I appreciate you coming on the podcast. It's been a wide-ranging tour of subjects.
Thanks for having me.
Thank you.
Yeah, sure.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.